When a neuroscientist claims a brain scan shows love, fear, or decision-making lighting up a specific brain region, what is actually being measured? Not thoughts directly, but blood flow, electrical voltage, or glucose consumption, each captured by a different imaging technology with its own strengths, blind spots, and interpretive pitfalls. Understanding what neuroimaging technologies actually measure is essential to understanding and appropriately doubting the flood of brain scan headlines in the news. Functional magnetic resonance imaging detects changes in blood oxygenation, the bold signal for blood oxygen level dependent, as an indirect proxy for neural activity, based on the principle that active brain regions require more oxygenated blood. fMRI offers excellent spatial resolution, typically 1 to 3 millimeters, allowing researchers to localize activity to specific brain structures with impressive precision. This makes fMRI the dominant tool for mapping which brain regions are involved in particular cognitive tasks, emotional states, or clinical conditions. Despite its spatial precision, fMRI has poor temporal resolution. The hemodynamic response, the change in blood flow following neural activity, takes 4 to 6 seconds to peak, meaning fMRI cannot capture the millisecond scale timing of actual neural firing. fMRI also only detects relative differences in activation between conditions, not absolute neural activity, and requires substantial statistical correction because scanning the whole brain generates thousands of simultaneous statistical tests, famously demonstrated by a study finding significant brain activation in a dead salmon shown emotional photos, when proper statistical corrections were not applied. Electroencephalography measures electrical activity generated by populations of neurons using electrodes placed on the scalp, providing millisecond-level temporal resolution, capturing the actual timing of neural events as they unfold. This makes EEG the tool of choice for studying rapid cognitive processes like attention shifts, language processing, and sleep stages. Its major limitation is poor spatial resolution. Because electrical signals disperse and blur as they pass through the skull, EEG cannot precisely localize which specific brain structures generated a given signal. Positron emission tomography involves injecting a small amount of radioactive tracer that binds to specific molecules of interest, glucose to measure metabolic activity, or specific ligands that bind to particular neurotransmitter receptors like dopamine receptors. PET is unique in its ability to visualize specific neurochemical systems directly, which is invaluable for studying conditions involving specific neurotransmitter abnormalities like Parkinson's disease or addiction. PET has poorer temporal and spatial resolution than fMRI, requires radioactive exposure, and is considerably more expensive and invasive. Reverse inference is the logically flawed practice of concluding that a specific mental state occurred because a brain region known to be associated with that state showed activation. The problem is that most brain regions participate in multiple different mental processes. Amygdala activation, for instance, occurs during fear, but also during novelty detection, positive arousal, and social evaluation. Concluding fear from amygdala activation alone, without other converging evidence, is a common error in both popular reporting and, historically, some published research. Neuroimaging technologies have transformed psychological science by making it possible to observe correlates of mental processes in living brains, but they are most powerful when combined with, not substituted for, careful behavioral and cognitive measurement. The strongest neuroscience research uses neuroimaging to test specific, theory-driven predictions derived from established cognitive models, rather than using brain scans alone to generate claims about the mind. Multimodal studies that combine neuroimaging with behavioral tasks, patient lesion studies, and computational modeling provide the most convincing evidence. Every neuroimaging technique offers a partial window into the brain: fMRI for spatial precision, EEG for temporal precision, PET for neurochemical specificity, and none provides a direct readout of thoughts or feelings. Understanding these tools' actual mechanisms is the difference between being persuaded by a headline and being an informed consumer of brain science.